I was chairing a session today on the earlier diagnosis of cancer, and particularly looking at the question about whether molecular diagnostic tests are ready to be applied clinically, and whether that could really improve outcomes for patients and aid clinical earlier diagnosis. So there were 3 speakers at the session. The first speaker was Dr. Marc Tischkowitz, who's also from the University of Cambridge, and he was talking about tests for genetic predisposition, particularly for ovarian cancer. So we've known for quite a while that patients who inherit, for example, a mutation in one of the BRCA genes, BRCA1 or 2, are at risk for breast cancer and ovarian cancer. So what he's been looking at is using some of the newer next generation sequencing models to test wider segments of the population, not just those people at the very, very highest level of risk, but people who have some family history who might be at increased risk. And he presented some preliminary results from a new study that he's been doing to see if that's really feasible, to see if that's economically viable, and also at the acceptability of it, because you have to, of course, address the question for patients whether this could increase their worry factor, whether it's actually really a helpful thing to do, or whether it makes people just more concerned. And the results of his study were extremely encouraging, so suggesting really that this is quite feasible logistically to do in clinical genetics on a bigger scale than we've been doing it before, and patients actually cope with it very well and found it generally acceptable, reassuring. And these patients have a family history of cancer, so they're very keen, of course, to reduce the risks for their other family members. And they've also got some preliminary data on health economics, which look encouraging as well. And then the second speaker was Professor Peter Sasieni from the Cancer Prevention Unit at Barts, and he really addressed a bigger question about molecular biomarkers and how they're useful at different stages, different tiers, if you like, in earlier detection. So both genetic susceptibility, as I was just mentioning, but also for finding precancerous lesions, and also for patients that present late, just trying to get them earlier. They've already got a cancer there, but they don't go and seek help because their symptoms might be quite low level. So he outlined some of the difficulties, the challenges, the controversies, how we need to approach this in terms of minimising overdiagnosis, minimising underdiagnosis, making sure we've got the right molecular biomarkers, but also the right sampling procedures. And that led on nicely to my talk, which was more of a specific case example, if you will, of esophageal cancer. And I was outlining the work we've done to develop a new diagnostic test for cancer of the esophagus, in which we've tried to embrace molecular biomarkers on the one hand, but also better cell sampling technique. So currently the gold standard diagnosis is endoscopy, and endoscopy isn't feasible on a kind of population scale for everyone with a bit of heartburn, which is the main risk factor. So we've been developing a GP-based test, very simple test, little pill on a string that you swallow down, pill dissolves to expand to give you a small foam sponge. You then pull it back and collect a lot of cells on the sample, about 500,000 cells. Then we couple that with a biomarker for Barrett's called trephor factor 3. So I was presenting the latest results of our research studies, which are very encouraging, and also how we can not only diagnose Barrett's, which is the precancerous condition, but identify which patients are already on their way to getting cancer who need treatment versus those that are at low risk, so that we don't just triage everyone into endoscopy. So that was some new data I was presenting on the risk stratification, and explaining that we think we need one more big trial before this is ready for GPs to use. And really, if you're interested in molecular diagnostics, you have to be in it for the long haul, because it's not a quick win. You have to be persistent and keep going towards your goal.